Okay, good afternoon, colleagues. We are just on time. It's the last session for this day, so it is my great pleasure to welcome you all to the second part of the session on transfer, incorporation of science into radiation protection system. This session will be chaired by Dominique Laurier, ICRP Main Commission member, chair of Committee 1, and a Deputy Director of the Health Division of the Institute of Radiation Protection and Nuclear Safety in France. And it is also chaired by myself, a Secretary of UNSCARE, United Nations Scientific Committee on the Effects of Atomic Radiation. And this is, I would say, a great opportunity to show how science and implementation of science in radiation protection go hand in hand. So we're both very excited and looking forward to hearing 5 presentations from expert teams from 3 continents about their experience, projects, and open questions on this topic. After this session, there will be a joint panel discussion on implications of new science on updates to the system of radiological protection. So please stay with us. This joint panel discussion will be held in about 30 minutes. It will take about 30 minutes from 5:15 until 5:45 today. So there's a little bit of a change with 15 minutes. So we would encourage you to keep the questions and interventions until the end of this session and to participate in the discussion and share your views, your feedback with all our panelists from this session and the previous session, the speakers from both sessions. So if time is not sufficient, please send any of your questions to Dominique and myself, and we'll make sure that we follow up and you receive the answers in writing. So without other delay, I would like to introduce the first speaker, and this is Mr. Ludovic Vaillant. Sorry for the pronunciation. He's an ICRP Committee 1 member and a project manager at the Nuclear Protection Evaluation Center in France. He's going to give a talk about the classification of harmful radiation-induced effects on human health for radiological protection purposes. This is a work that is being carried out by a dedicated ICRP Task Group 123. This group is chaired by Ludovic. So you have the floor. Thank you.
So, thank you very much for the kind introduction. Thank you to the organizers for inviting me and to make this presentation. So, as an introduction, to make sure we are all on the same page, a couple of definitions. I think you're all pretty aware of this definition, but the current classification of the system considers 2 types of effects. Tissue reactions, also termed deterministic effects. A tissue reaction is an injury in population of cells characterized by a threshold dose, and an increase in the severity of the reaction as the dose is increased further. We also consider stochastic effects. Basically, stochastic effects are cancer and heritable effects. Those effects are effects resulting from damage in a single cell. The frequency of the event, but not its severity, increase with an increase in the dose. For protection purposes, it is assumed that there is no threshold dose. This definition is taken from ICRP. So, if we move now to the objective of the system of radiation protection with regards to the protection of human health, this is stated in publication 103. The objective of the system is simple. First, harmful tissue reactions are prevented. Harmful is an important adjective there. And second point is that the risk of stochastic effect are reduced to the extent reasonably achievable. So when you have a look to the definition, when you have a look to the objective, you understand that finally the classification of health effect really underpins the implementation of the system of radiological protection. Speaking of the implementation of the system, basically, it's pretty simple. With regards to the management of stochastic effect, this is the left side of the slide. Basically, we rely on the principle of optimization of radiological protection, keeping exposure, the magnitude of individual dose, and the number of exposed people as low as reasonably achievable. When we move to the management of and the prevention of tissue reaction, this is a graph there on the right side. You have a threshold dose. This is the red line we have there. You set up a limit with a margin of safety, and if you respect the limit, you are sure you're not going to exceed the threshold, so tissue reactions are prevented. This is pretty much clear and pretty much simple, finally. And it works well. I mean, the system is robust. It provides good results. So I think that now you are all pretty much familiar with this paper, which is basically a roadmap for the next decades, keeping the system fit for purpose. Within this paper, there is this a paragraph which says that the classification of harmful radiation-induced health effect into stochastic effect and harmful tissue reactions for protection purpose should be revisited to ensure that it remains fit for purpose. A couple of examples are given there. We speak about cataract, we speak about DCS. Disease of circulatory system. So why are we speaking of this system and why they don't fit clearly into a current classification? So with regards to cataract, I took a scheme which is taken from publication 118. Publication 118 is dealing with tissue reactions. It is also in this publication that you have the ICRP statement that lead to the change of dose limit for the length of the eyes. The blue line there shows you the evolution of the numbers of opacities with the dose. It is a nice line. There's no threshold, but an opacity of the lens, it's not an harmful tissue reactions. So harmful tissue reactions, it is a cataract. And this is what the author plotted there with a nice red curve. And what you can see is that the cataract is occurring only after a certain level of dose. So within publication 118, the cataract is considered as a tissue reaction with a threshold dose of 0.5 gray. We had a discussion last Saturday within task group 123 and a couple of very nice presentations. One was from Liz Ainsbury. Hi, Liz, I think you are somewhere in the room. I asked her if I can use one of her concluding remarks. And speaking about cataract, at the end of her presentation, this is what she mentioned. Some new data on cataract detection and classification indicate that early lens change may be better characterized as tissue reactions with a clear threshold, whereas longer-term change are better described as stochastic. So cataract, stochastic, tissue reaction, something different. If we move now to the disease of circulatory system, DCS, here is a paper from Mark Little. So we move now to the side, epidemiology. For low dose and moderate dose, we've been studying the occurrence of non-cancer effects. Leading to the conclusions, I am myself not an epidemiologist, but what you can read is, we recommend statistically significant excess risk of the major types of circulatory disease, especially ischemic heart disease and stroke in moderate or low dose exposed group, with some not altogether consistent evidence suggesting dose response non-linearity. So some question there. Is it a stochastic effect or is it a tissue reactions? So you understand the need finally to visit again the classification of health effects. So before going to the mandate of task group 123, I wanted to use, so I'm really sorry, it's late today, it's a lot of text on the same slide, but this is an excerpt from publication 9 from 1966, general recommendation of the Commission. I wanted to shorten it, but finally every word is pretty interesting and matters there. What the authors of the publication wrote in there is the mechanism of induction by radiation of leukemia or other type of malignancy, we're talking only about cancer there, is not known. Such induction has so far been established only after dose of more than 100 rats, basically 1 sievert or 1 gray. They don't know at the time if there is threshold dose. But what they do is, the existence of a threshold dose is unknown. It has been assumed that even smallest dose involve a proportionally small risk of malignancy. Also, because of the lack of knowledge of the nature of the dose-effect relationship in the induction of malignancy in men, the Commission sees no practical alternative for the purpose of radiological protection to assuming a linear relationship between dose and effect, and that those act cumulatively. The Commission is aware of that the assumption of no threshold and complete additivity of all those may be incorrect, but is satisfied that they are unlikely to lead to the underestimation of risk. We've been talking a lot about values, ethics, and when I read this, not being an expert like Kunwo Shaw with regards to publication 138, but what I see there is prudence. What I see there is non-maleficence. What I see there is accountability. So I think it's really interesting to try to understand or to have this question in mind. What the authors of this publication would do today with regards to DCS or cataract. Of course, our answer is not straightforward because science is more complex, society has evolved, values and so on has evolved. But it's really interesting to keep this in mind. So let's go now to task group 1, 2, 3. It's a newly created task group, and basically we have a first meeting last Saturday afternoon. We have 3 objectives within this task group. The first is to clarify the rationale behind the current classification. So it's not only about giving the definition and the objective of the system. Is to try to understand how this classification has evolved with time, why it has evolved, and what is the link between the classification and the objective of the system. So our plan there is to get back to publication 9, 26, 60, 103, 118, and maybe others, to try to clarify this aspect of the system. We talk a lot about rationality of the system, I think this morning, also about clarity, and I think it's really important to get back into this. The second objective of the group, what are the reasons calling for an evolution or not? And this is will be based on a review of scientific literature. There we are not intending to make a systematic review of the literature like the UNSCA. We are going for what Chung Cheng Li, who is a member of the group, called a narrative review of the literature. We have experts in the field in the group. They are going to identify the key papers, and we are going to elaborate on these key papers. And based on this task 1 and task 2, basically, we are going to try to see if there is a need for an evolution of the system. And if there is a need for an evolution of the system, what are the consequences on the practical management of radiological protection? So here I put this little graph because I think that within TG 1, 2, 3, it is really important that it's not only about science, it's also about experience, it's also about values judgment and what we call the ethic of the system. So I don't want to frighten you. I pick these slides from a presentation by Chris Clement. The system, as it is today, is performing well. It is robust. However, we need to take into account that science is bringing new evidence on the flow, and we need to assess the consequence of this new knowledge on the system, and we need to make sure that the system remains fit for purpose. Another aspect of our work is that I think Werner mentioned that we have kind of 30 task group ongoing within ICRP. We need to make sure we work in a consistent way. So here I mentioned a couple of task groups. You have task group 115 dealing with risk and dose assessment for astronauts. You have task group 118 dealing with disease of the cardiovascular system. Task group 121 dealing with exposure in the offspring, which was mentioned earlier today, and also task group 122 dealing with the update of detriment with regards to cancer. It's really important that we are consistent within our work. What was really interesting in, I think, the question by François Bouchu to Richard Wakefield is, for instance, hey, it is true that this 10 milligram to the, as an intraocular exposure can lead to some cancer. Apparently, yeah, and we need to consider this with regards to classification of health effects. I mentioned here also for a co-chair that there are also kind of train or sister group within the UNSCA working on some topics which are pretty much similar but than some of the task group of ICRP. We are working on the same topic, but we don't have the same objective in mind. Still, for the sake of clarity, it's really important to work together and to make sure we are consistent within our work. Task group 1, 2, 3, finally, is not only a committee 1 task group, it's also a committee 4 task group, because as I mentioned, it's not only about science, it's about the application of the system. So finally, to conclude, here is a list of the members of the task group. I really want to thank them for the commitment to work for the task group. We had a great meeting last Saturday. Special thanks to Liz Hansberry with a vice chair, to Frido Zolzer also with a vice chair, and also special thanks to Konstantinos Servodes, a technical secretary who is doing a great job. He was appointed by the Commission and he is really doing a great job. And finally, thanks to you for your attention.
Thank you very much, Mr. Valiant, for the very informative presentation, and also I'm sure that the radiation protection community will follow up the progress of this group very, very closely, and thank you for also providing the link and showing the link between INSCARE and ICRP. So I think we've agreed with Dominique that for the interest of time, and there's going to be a joint session after that, we will move on to the next presentation. And our next speaker is Mrs. Christelle Goulerbin. She's an ICRP Committee 1 member and a leading radiobiology research program. She's leading these programs at the microbeam of the Institute of Radiation Protection and Nuclear Safety in France. She will talk this time via pre-recorded video. Unfortunately, she could not join us. And she's going to talk about the relative biological effectiveness for reference animals and plants, a topic in the scope of ICRP Task Group 118.
Good afternoon, ladies and gentlemen. I would like to thank the chairperson for inviting me, and I apologize for not being here to present you this overview on RBE and radiation weighting factors for reference animal and plants. As you may know, the risk of radiation exposure does not only depend on dose and on dose rate, but also on the characteristic of the radiation causing the dose. To take that into account, the concept of relative biological effectiveness has been introduced several years ago. So RBE is a ratio of a dose of low linear energy transfer reference radiation, usually X or gamma rays, to a dose of a test radiation considered that gives the same level of a biological effect. RBE depends on dose rates, dose fractionation, endpoints, cell types, species. So, due to the complexity of the parameters to take into account for RBE, it was proposed to restrict its use for radiobiology only. Another quantity was proposed for an application in the field of radiological protection. It's the quality factor, which can be used to weight absorbed dose to obtain a common scale for all ionizing radiation. So over the years, the concept of quality factor has been modified, and the last major evolution was made in the 90s in publication 60, where ICRP introduced a modified concept. It was proposed that the absorbed dose is averaged over an organ or tissue, and that this absorbed dose average is weighted for the radiation quality by WR, the radiation weighting factor. So different values of radiation weighting factors have been proposed, and the last WR re-evaluation was done in 2003 in publication 92. So you can see on this table the different values of WR. These values were of 1 for photon, electrons, and muon, of 2 for protons, of 20 for high-lead radiation, and from 5 to 20 as a function continuous of energy for neutrons. So how do these concepts apply for environmental radiological protection? Indeed, the foundations of environmental radiological protection system have been laid in publication 103 and 108. In particular, particular, the targets of protection have been established. They are the maintenance of biological diversity, the conservation of species, the protection of the state of natural habitats, communities, and ecosystems. So we deal with a population level mainly. Furthermore, due to the immense variety of species in ecosystems, key reference elements had to be defined. Hence, 12 reference animal and plants, RAPs, have been adopted, which are typical of main ecosystems. These RAPs can hence be used for relating exposure to dose and dose to effect. Additionally, derived consideration reference levels, DCR, were also adopted in order to consider the risk. These DCRL correspond to the different RAPs, and DCRL are bounds of dose rates within which there is likely to be some chance of deleterious effects occurring to the considered RAP. Those levels can be used as a point of reference to optimize the level of effort expended on environmental radiological protection. So it can be underlined that no radiation weighting factor was recommended in this publication. So it was the point of publication 148 that was published in 2019. The aim of this publication was to review the data available on RBE for tritium beta particles, chosen as an important example of low energy, low lead radiation, and for alpha particles. The aim was to propose weighting factors that can be used to calculate absorbed dose rates for the different RAPs. So the biological endpoints that were reviewed for RBE are those of most relevance to the protection of non-human biota. In other words, they were those that could lead to changes in population dynamics. They include survival, fecundity, reproductive and developmental impairments, which are deterministic endpoints. Other endpoints, such as DNA damage, chromosomal aberration, mutation, and tumor induction, stochastic endpoints, have not been shown to be directly linked to population dynamics. However, for completeness and to allow comparison to be made, they are included together with directly relevant data on tissue reaction. Thus, biological data are grouped for analysis in 4 categories: mortality, reproductive dysfunction, morbidity, and chromosomal damage. Data on cancer induction are included in the morbidity category. You can see on this slide a summary of the main feature of tritium RBE, with one example of a graph showing the distribution of RBE for reproductive success. The 3 bands of color represent the DCRL for rat, fish, and worms, 3 of the RAPs. Most of data come from mammals or cell lines, with only a few fish and invertebrate data. In addition, exposure to tritium was made using intraperitoneal injection, which is not very representative of exposure in the environment. RBE were calculated for dose rate higher than DCRL, and the values range from 1 to 3.9, and they were lower when X-rays were used as a reference radiation than when gamma rays were used. You can observe on this graph a summary of RBE for alpha particles with 2 presented graphs. On the left, it's the RBE distribution for reproductive success, and on the right, RBE distribution for chromosomal damage. So here again, a majority of data come from mammals with only 1 fish value over 68 RBE value. The RBE were calculated for those rates often higher than DCRL, and they range from 1 to 20 and were centered at around 10. So the main points of the publication are double. So the first one is that RBE weighted absorbed dose rate to RAPs should be calculating using a value of 1 for low lead radiation and a value of 10 for alpha particles. And the second conclusion is that a caveat is made if exposure to tritium are within or close to the DCR band. In that case, an additional review of RBE weighting should be required. Another task group has been launched recently by ICRP. It's the task group 118, which aims at reviewing data on RBE, quality factor, and radiation weighting factor. In particular for humans, since these data have not been reviewed since publication 92 in 2003. This task group is under C1 and C2, and in this context, it was also decided to review RBE values for the environment as a supplement of publication 148. So different papers were reviewed. So the work is still ongoing, but it was possible to find additional papers on RBE for wide species. So for tritium, RBE of 3 were calculated for tritiated water in several aquatic organisms for the endpoint of reproduction. And for strontium 90, for the plant, an RBE from 1 to 3 was also calculated for growth. For alpha particles, a review of several papers on mammals has proposed a generic weighting factor of 15 for all alpha particles, and a specific value of 50 for plutonium and americium, and of 5 for radon. Interestingly, weighting factors have been shown to increase with lifespan. In addition, other RBE have been calculated from 3 to 10 for americium in different invertebrates and fish species for reproduction, and also a conservative RBE of 35 was proposed for polonium for the reproduction of fish. As publication 148 was focused on tritium and alpha particle, it was also decided to review papers related to other types of ionizing radiation, and in particular for heavy ions, neutrons, and protons. So this type of ionizing radiation may be interesting for the context of space radiobiology, and for non-human biota, this can be also of interest because several of these species are studied as a candidate species for food autonomy of human space colonies. In addition, those models provide different transients and mutations that allow to investigate different radiobiological mechanisms. So for heavy ions, I'm not going to go into detail, but the highest RBE value was found for carbon ions at a value of 52 for the lethality of higher plants. For neutron, the highest value was at a value of 10 for the development of zebrafish, and for proton, the value of 2.5 was evaluated for development of zebrafish. To conclude this presentation, it can be said that RBE data for non-human species is still lacking, in particular for high-rate radiation. However, additional values can be taken into account for tritium and alpha particles. Radiation weighting factors may also be proposed for protons, heavy ions, and neutrons. Plant RBE seems to be higher than animal RBE, with a value of more than 50 for topicocide and carbon. This underlines the interest of studying different species to better understand the diversity of biological mechanisms involved in RBE. RBE calculation methods need also to be analyzed, and in particular for moderate to high dose rates encountered in the environment. The issue of target size is also highly important, and different targets may be considered organ, cell, nucleus, DNA, cytoplasm. Finally, recent advances in radiobiology, radioecotoxicology, dosimetry need to be included in radiological protection. For example, non-targeted effect, molecular biology data, microdosimetry studies. For that, a close collaboration between radiobiologists and dosimetrists is needed. I thank you very much for your attention.
Although Christelle Adam is not here, I would like to thank her for this very, very comprehensive presentation on a topic that is very relevant also for UNSCARE, which is considering also this topic for its future program of work in the period 2025-2029. And allow me to pass the floor to my co-chair, Dominique.
Thank you very much. So with no transition, I would like to invite the next speaker. So the next speaker is Nobuo Ikoban. Nobuo Ikoban is a commissioner at the Nuclear Regulatory Authority in Japan, but is also a member of the ICRP Committee 4. And more importantly for this presentation, he has been the chair of ICRP Task Group 102, who has been working on the methodology of calculation of the detriment, and is going to present some conclusion from this task group. So Nobuo, the floor is yours.
Thank you, Chair, for the kind introduction. I'm going to talk about the past, present, and possible future of detriment. The most recent ICRP publication is publication 152, Radiation Detriment Calculation Methodology. My talk is mostly based on this publication, but not exactly the same. It has my flavor. My talk consists of 3 parts: historical development, potential evolution, and final words. Let me start with the first part. The concept of detriment was first introduced in publication 22 in 1973. That means detriment concept has a half-century history. It was defined as the mathematical expectation of harm as shown in this formula. Initially, the harm was intended to encompass all sorts of deleterious effects, including the socio-economic impact. However, only health effects were considered in practice, so now detriment is regarded as an expected value of the weighted number of health effects. This slide summarizes some points of detriment concept. First, it was defined as the expected value for a group, not for an individual. It considers the probability and severity of effects as shown in the previous slide. It is intended to be used for low dose and/or low dose rate exposures. Therefore, only stochastic effects are considered. Detriment concept was redefined as a multidimensional concept in publication 60. This means we can assess detriment using various metrics such as lifetime risk or years of life lost. But they express just only part of the aspect of the detriment. Detriment itself as an abstract concept. But still, detriment as a single index continues to be used in particular for the purpose of determining values of tissue weighting factor. So this detriment as a single index is specifically called radiation detriment. Radiation detriment should not be considered as a projection of the absolute number of cases in an exposed population. It is just an inference based on assumptions to assess the potential health impact for radiological protection purposes. This table summarizes historical transition of radiation detriment in ICRP recommendations. In publication 26, detriment was quantified as so-called risk factor. It is a probability of fatal cancers and severe hereditary effects, but non-fatal cancers were not considered. In publication 60, detriment was expressed as a weighted number of cases of cancers and serious hereditary effects, and number of cancer sites was increased to 12 individual organs or tissues and other solid cancers. In publication 103, the calculation was further elaborated. Incidence-based cancer risk model was used, and risk of heritable diseases were reassessed. For severity adjustment, QOL factors was newly introduced. This figure shows the procedure for calculating radiation detriment in publication 103. Calculation of radiation detriment consists of mainly two parts. Nominal risk calculation. Nominal risk is lifetime risk estimate for a nominal population averaged across sexes, ages, and geographical regions. And the second step is severity adjustment. I do not go into the detail of these calculation steps, but what I want to emphasize in this figure is calculation of radiation detriment requires many steps and considerable number of data set. So these are past and present. Now let's talk about possible future. So what should be done for the future? There is some room for improvement. I categorize the room for improvement into 4 items. First, improving quality of estimate, handling of variation, transparency, and communication. To improve the quality of estimate, first of all, it is necessary to incorporate recent scientific findings. It includes re-evaluation of DDREF. This does not mean just a choice of the value for DDREF, but to evaluate the dose response for low dose and low dose rate exposure. Cancer risk model used in publication 103 are based on an epidemiological study of atomic bomb survivors as of 2007. And risk estimate of heritable diseases is based on UNSCARE 2001 report. So these models and estimate need to be updated. Consideration of non-cancer effects. This is one of the hottest topic, as Ludovic presented. Particular, whether to include circulatory disease in detriment calculation. It is a controversial issue, but we need to address. Calculation of radiation detriment requires baseline statistics and demographic data. For publication 103, data of 1990s were used, so they must be updated. And also, for adjustment, so adjustment for severity, these parameters are based on older data as well, so it should be updated. Now let's talk about variation. This figure represents radiation detriments for the whole population in publication 103. Let's take a detailed look at these three tissues. They are three highest values of detriments. So that is lung, breast, and other solid cancers. This slide shows risk coefficients, in other words, lifetime risks calculated by the method described in publication 152 for these three tissues. Left side panel is for male and right side for female. For lung, you can see the curve is almost flat except at older ages. But for others, you can see the quite high lifetime risk for young age at exposure. And for breast, risk is assumed to be 0 for male, so significant sex difference. There are many sources of variation. In addition to sex and age, genetic background, lifestyle factors, and medical resources, because it depends on fatality of cancers depends on medical resources. Question is how to handle variation. One way is to use a single value as a worldwide average. Another way is to divide into some subgroups. For example, in the current case, radiation detriment is calculated for the whole population and working age population in two ways. So calculate average for each subgroup. That is another way. And of course, there is another possibility, fully individualized approach, although this is quite resource demanding. Whatever the approach, there is pros and cons, but we need to make a decision. Which approach is better, maybe depending on the situation. Transparency. Calculation of radiation detriment is a complex process. So first, we need to clarify information sources. And also, it is vital to document calculation procedures. Unfortunately, miscalculation was found for detriment calculation of working age population in publication 103. Not to repeat this kind of mistake, cross-validation of calculation is quite important. Ultimately, it is desirable to develop open source software. And finally, communication. Currently, radiation detriment is expressed as a weighted number of cases of cancer and heritable diseases. But it is quite difficult to the general public what these numbers mean. So it may be time to reconsider the way of expressing detriment. One possibility is to adopt a metric widely used in other fields, such as disability-adjusted life years DALY. And it will be helpful to graphically represent key components of parameters and data used for detriment calculation, and if possible, visualizing the calculation process will be better. And calculation of radiation detriment is based on assumptions, and data includes uncertainties. We need to convey the source and degree of those uncertainties, but it is another challenging issue. Final words. In summary, radiation detriment should be based on sound science, should exercise prudence where uncertainty remains, should not be overly complex, and should be comprehensible. This means makeup of radiation detriment reflects our wisdom as well as scientific knowledge and understanding. So I want to conclude my presentation with a quote of Pericles, a politician and general of ancient Greece. He said, "Having knowledge but lacking the power to express it clearly is no better than never having any ideas at all." Keeping this quote in mind, we should do our best in this challenging task. Thank you for your attention.
Thank you very much, Nobuhiko. It seems to me as a very wise view on the construction of detriment, and it will be very helpful for the evolution in the future. I would like now to invite not only one speaker, but two speakers from Canada for the next speech. So Christy Rondawa, who is a Radiation and Health Sciences Officer at the Canadian Nuclear Safety Commission, and also recently joined the Secretariat of the UNSCARE as a part-time in-kind expert. And the second speaker is Ruth Wilkins, who is the chief of the radiobiology division at Health Canada. So Christy and Ruth, the floor is yours.
Thank you, Chair. It's a pleasure to be here, and thank you all, or appreciate you all for being here near the end of day 3. I am Christy Randawa from the CNSC, and I'm joined by Ruth Wilkins from Health Canada, and we are the current coordinators for the Canadian Organization on Health Effects from Radiation Exposure, or COHERE. So a brief outline of what we plan to go through today. I will provide a brief introduction to our fairly new program, go over the vision and objectives, provide an overview of our structural framework, our strategic research agenda, and then I will pass it on to Ruth to highlight some of our projects and speak about some of the outputs or intended impacts of the program. So COHERE is a joint initiative between Health Canada and the CNSC, which are both federal agencies of the Canadian federal government. Both agencies conduct and coordinate research on health effects from radiation exposures, and both agencies recognize the importance of collaboration and coordination of research on important topics of interest to stakeholders. So this initiative is directed towards a better understanding of health effects or health risks from exposure to low doses of radiation, and can also be used to inform UNSCARE as well as impact risk assessment. So COHERE's vision is to contribute knowledge to reduce scientific uncertainties from low dose and dose rate exposures, and to advance our understanding by bridging the knowledge gap between human health risks and linkages to molecular and cellular level responses to radiation. And to accomplish this, we need aligned research priorities between Health Canada and the Canadian Nuclear Safety Commission in order to leverage resources. We need expertise through maintained and enhanced expertise in dosimetry, radiobiology, and dosimetry, and informed and consistent messages to the public as well as stakeholders. Currently, the structural framework is comprised only of representatives from Health Canada and the CNSC to better align our research priorities through these organizations. So as mentioned before, Ruth and I are the program coordinators, so we coordinate and promote COHERE's activities. Our core members are the scientific committee, and these are the individuals who conduct, present, and publish research. And these are the individuals who we worked with to establish our strategic research agenda, as well as our work plan. We also have our very important champions, who are management from our organizations, who also support and promote COHERE. And we have a communication committee who help to develop our communication plan, provide website support, as well as design any communication materials that we require. So our strategic research agenda currently has 5 themes, which are broken down into research lines and subsequently into priority areas. It's quite complex and is a very large table, so it is available on our website, and I'll provide some details on that on the next slide. So our 5 themes are currently cancer effects, non-cancer effects, globalized data sharing and consolidation, capacity building, as well as epidemiological studies. So these priorities were identified by the international community and other research platforms, including the ICRP, as well as MELODY, which is the European platform for low-dose research. And our SRA aims to address challenges and inconsistencies that are encountered when applying and updating current radiation protection regulations and recommendations, as well as when communicating to stakeholders. As noted before, our SRA also considers stakeholder input, so these can be public inquiries as well as concerns, which as federal government entities we tend to receive. So the strategic research agenda priority areas narrow the scope to focus on topics of primary concern in the Canadian landscape, as well as opportunities, tools, technologies that best leverage the Canadian federal expertise. So now I'm presenting our webpage. So it is hosted on the CNSC website and provides an overview, some objectives, as well as our projects. It's currently in the works of being updated for 2022. It also highlights our researchers who are currently involved in the project. And there is a link for our strategic research agenda, which provides more details on these projects and goes into the details of our research lines as well. So I will now pass it on to Ruth, who will highlight some of our current work.
Thank you, Christy, for that great introduction to COHERE. We coordinate not just our research, but we also like to coordinate our presentations. So let's see if we managed well. So to continue on where Christy left off, this is just a list of some of our priority questions. We have several, but we just want to highlight a few. So we're asking questions such as, how does chronic exposure to radiation lead to disease progression, and what are the key events and interconnectivities? Is the distribution of radionuclides and other subsequent energy deposition inside the cell or tissue homogeneous? And which biomarkers can indicate biological effects of low dose and low dose rate exposures to ionizing radiation, and how can they support risk assessment activities? So as Christy mentioned, we have a website, and if you want more detail about ongoing projects, you can go to that webpage, and you can see a list of all the ongoing projects. And as Christy mentioned, we are in the process of updating that, so there will be new projects being added shortly to this site. We also have been quite successful at publishing some of our work since we started COHERE, and we have developed a research page or project where you can go and see all of the publications that have come out of our collaborations. So just to give you a spotlight on our activities, We have our terms of reference and strategic research agenda that Christy had mentioned. We now have 17 projects on our website, but that will be growing. We have published our seminal paper, our vision paper, which outlined all of our visions, our strategic research agenda, some of our partners and projects, and international work that we've been doing. And our next big item on our agenda is to try to coordinate low-dose research across the country, so involving other partners, which I will talk about in a moment. So for national coordination, our goal is to pull together not just the federal partners who are doing low-dose radiation, but also pull in government and industry. So this is really being spearheaded by COHERE, so Health Canada and CNSC, but we are also bringing in the Can-Do Owners Group and CNL, ACL. We have representatives from all of those organizations here today, which was very useful because yesterday, at lunchtime, we gathered together to start to discuss what our next steps will be in national coordination. So the motivation of national coordination is to build upon the existing low-dose radiation research groups in Canada, and this will enable a structured coordination of the Canadian research program. And the benefits or results of this will be enhanced collaboration, it'll improve knowledge transfer and communication, which will enable sharing of ideas, will prevent the duplication of projects within the Canadian landscape, will increase the efficiency of use of fundings and sharing resources and facilities and identifying priority research. This will also allow us to coordinate as a group to submit proposals to funding agencies to leverage some of the expertise between the different groups and generate large project proposals with hopefully a better chance of receiving funding. And then also be more effective in supporting the international efforts. So we would like to gather and coordinate our low-dose research at a national level to help feed into organizations like the OECD high-level group on global coordination of low-dose research. So in summary, COHERE benefits the radiation protection community by broadening the international profile of Canadian scientists and their research laboratories. It's demonstrating leadership in the radiation protection community within Canada. It's creating a point of contact within the Government of Canada for more effective coordination and collaboration with the national and international organizations. And results from COHERE will continue to contribute to the international protection framework. So there's a body of evidence that informs the international radiation protection framework. So for example, we have people working from COHERE who are involved with the United Nations Scientific Committee on the Effects of Atomic Radiation, both as representatives for our country. Also, we have some individuals who are doing in-kind contribution to the UNSCEAR Secretariat. We have involvement with ICRP, both as members, task group members. We have mentees within ICRP as well. We've also done some work with the International Atomic Energy Agency with some of their coordinated research projects. Also working as consultants for the IAEA. We also have members involved with the OECD high-level group and of coordination of low-dose research. So we have a very strong group of Canadians in this field who are getting involved on the international level. So also we will have the application of incorporation of international recommendations into Canadian regulations and into national guidance. So if you have any questions at all, please come and see Christy and myself at this meeting tonight at the gala. We'd be happy to answer any questions. This is a list of our scientific committee. So these are the scientists who are involved in doing the research for COHERE at this time. You can visit our webpage. You can look at our ResearchGate project. And our contact information is there if you'd like to reach out to us by email. So thank you very much.
So thank you, Christy and Rose, for this very good presentation of the COHERE research program, which appears as one of the main projects of research in the low-dose area. And I really think it's important to have a good coordination of this project with the other ones ongoing in the world. I'm thinking about the pianoforte, especially in Europe, for example. So we are now moving to the next and last presentation, which will be given by Nicole Martinez. So Nicole is an assistant professor at the Clemson University in the Department of Environmental Engineering and Earth Sciences. And she's also the vice chair of the ICRP Committee 4. And in addition to that, she's the new chair of a new task group, ICRP task group, and that's the topic of our presentation right now on ecosystem services in environmental radiological protection. So, Nicole, the floor is yours.
Great, thank you so much. Thanks everyone for being here. Today I'd like to give you, because I think it's probably new to a lot of people, just kind of an overview of ecosystem services, what it is, and also a motivation for why we are starting to look at it. So one of everyone's favorite papers now, just in case you don't have it bookmarked, the fit for purpose paper, there's the QR code. But it highlights how we've kind of approached protection of the environment thus far. Largely, we focused on a conservation approach, which is fine. That's great. There's nothing wrong with that. But maybe there's more to it than that. With this conservation approach of the natural environment, maybe we need something more robust to also consider resources and cultural value of our environment. So ICRP publication 91 talks about conservation, preservation. It talks about ideas like the precautionary principle. But largely when we've talked about protection, it's been for non-human biota and hasn't really thought about resources, or how we've thought about resources, but like how we actually incorporate that. Ecosystem services in general and environmental protection in general has gained in popularity over the past 10 years or so. A large part of that is associated with environmental degradation and climate change and this interest in sustainable development. So in radiological protection, of course, we want to maintain that appropriate level of protection, but we also don't want to unduly limit human actions or interfere with sustainable development or come to unintended consequences, which we've seen in the past and ideally we try to avoid. So ecosystem services is one tool or approach to consider that. So what are ecosystem services? In general, in the top orangish box here, benefits we derive from the workings of the natural world. So any benefit we get from our environment. That underpins our economy. It promotes our physical health and also our well-being. So ecosystem services has the potential to help us take a holistic look at environmental radiation protection. Ecosystem services itself is not new. This idea or concept has been around since probably the 1980s, but it started gaining in popularity with the report you see here on the screen, the Millennium Ecosystem Assessment. And people are doing this, and I just have a quote here for our regulators or our practical people. Although not perfect, the suite of ecosystem services classified by the Millennium Ecosystem Assessment provided helpful for comparing benefits of different protections, is what that document goes on to talk about. So just to kind of persuade you, you don't need to look at all the small text here, but look at all the arrows. There are so many different categories of well-being that we could consider, and ecosystem services contributes in many ways to that. So that's the idea of this slide, is that ecosystem services really underlies our health and well-being, and that's one of the things we've been thinking of, right? Like, so what do we mean by health and overall well-being, and how do we approach that? Ecosystem services is kind of subdivided into four, sub-services, I guess you could say. Provisioning is probably what most of us think of when we think of like benefits we get from the environment. This is like the stuff, right? The direct products of the ecosystem. Who had salad today for lunch? Yeah, ecosystem service. That food came from our ecosystem, right? Our drinking water, a lot of building materials, fuel, there are all these things, penicillin, that's pretty great, right? So all these things that come from our environment will be considered provisioning services within ecosystem services. Supporting is probably is the most foundational, perhaps one that we might kind of not think about. It's so simple, but it's everything that keeps the ecosystem going. So photosynthesis. If you don't have primary production, you don't have life, right? It's really important. The nutrient cycle, the water cycle, all those things are considered supporting ecosystem services, the things that sustain us. Then we get into regulating, so things that moderate the natural environment. We have pollination, we have decomposition. You could have, in South Carolina, the seagrass helps mitigate flooding. Along the rivers, you have things that mitigate flooding. We have carbon storage, things that help with climate regulation. One of the things with habitat destruction that we're so concerned with is losing these carbon sinks, right? And that could potentially contribute to increased carbon in the environment. So regulating are also a very important ecosystem service. That brings us to cultural. These would be our non-material benefits. One of my favorite museums is L'Orangerie in Paris. I'm sorry for my pronunciation. But this museum has Monet's water lilies as these beautiful, long, panoramic pictures, and they have these meditative rooms. And I like it because I go there, and it makes me feel like I can remember the beauty in the world. It makes me think about how to find beauty in the world. And that comes from a cultural ecosystem service. Monet was inspired by water lilies, and I am inspired by Monet. Right? So this is considered a value of our ecosystem. How many people have gone for like a walk or a run outside? Oh, everyone's indoorsy. Okay, some people are outdoorsy. And thought about the, one thing I noticed when I was out was the color of the leaves, like how pretty the fall leaves are, or how blue the bay is, or how nice the mountains are with the dusting of snow. Yeah? Just me. Okay, some nods. Yeah, so all that is also a cultural ecosystem service. If you go sailing, that would be a cultural ecosystem service. And also there's more than that too. There's also various cultures have sacred pieces of this bit as well, right? So all of that would fall under non-material benefits associated with our ecosystems. Okay, so how many people think that ecosystem service is probably pretty good? Probably need food, probably like art, enjoy the fall leaves. So ecosystem services, yes, we need them, right? But the critiques and challenges of the use of ecosystem services come in how they're applied or how they're used in practice, okay? So I want to go through a couple of those. I think they're really important to go through and also offer some counter arguments. Some of these are interrelated, but they largely fall under these three broad categories. The first, I will say, is environmental ethics. So because it is the benefits people derive, that's anthropocentric, meaning human-centric. And so we've kind of moved past that, right? We've started to recognize that there is inherent value to nature. So we would call that the integrity of nature that Frido mentioned in his lecture. And so if we just think of ecosystem services, we could say, oh, that's very anthropocentric. And so maybe that's a regressive way of looking at things. And that also could potentially then be exploitative of nature. However, the proponents of the use of ecosystem services argue, well, it helps us find a way to compare things. So if someone like, I don't care about the environment, that's, you know, I'm in the US, so like that's the libs problem or whatever. But this is a way to say, no, it does matter. And here's a comparison I can show you. If you don't care about the integrity of nature, probably you care about the quality of your food, right? So some of the arguments are of that. They also say that ecosystem services still supports arguments for conservation and biodiversity in the benefits that we get from it, even if it is framed in a human-focused way. And they also argue, particularly with the cultural ecosystem services, that you could still help people reconnect with nature, so maybe to help people move past this purely anthropocentric approach. Another criticism is the economic framing. So the argument there, or the criticism, valuation could lead to selling out nature. For example, if I try to do an economic evaluation of my ecosystem, let's say if it's a wetland, and I say this wetland is worth $1 million, and then someone who has a lot of $1 million comes and says, here's $1 million, I'm gonna buy this wetland from you, and then totally destroys it, right? That feels kind of weird, right? Ugh, yuck. But so the criticism of this, in this putting things in terms of a dollar amount, could lead to situations like that. And there was actually a situation in Florida in the United States where something like that did indeed happen. So the counter argument to that is monetary valuation is not the only type of valuation. Kind of like we say value of statistical life isn't the actual value of an individual person's life. Right? It's just a way we have to be able to compare things because we have to have some unit of comparison in making these value judgments. But there are other ways you could do valuation, and this also could help raise the importance of it. So expressing something as even if it is monetary, to say this is how much it costs to, you know, I don't know how to do economic evaluation, but, but that can help raise awareness of how important it is. And then also the approach. This criticism is similar to the criticism of the precautionary principle in that the definitions are vague, and it's also very idealized. Like in a perfect world, this is how it would be, but that's not the real world, right? But the counter argument to that is that there's a lot of flexibility in this approach. So if you wanted to do a monetary evaluation, you could. If you wanted to do a different type of valuation, you could. And so it really supports that. And it also supports interdisciplinary collaboration. So not just social scientists or economists or folks who do environmental protection and ecologists, but all those types of people can work together to develop an approach that works for your specific situation. And then the other counter argument was basically, it's not wrong to have a positive attitude. What could be wrong with that? So I don't know how good of an argument that is, but that's one of the counter arguments for a positive outlook. So with all that being said, Task Group 125 is one of our new task groups, is going to take a look at ecosystem services. So how have people used it in other areas of environmental protection? Can it help support this holistic approach? And in that, how does that tie then back to sustainable development and the sustainable development goals, like Andy talked about, I think that was yesterday. So looking at definitions, also, we really want to make sure we look at practical examples. So a large bit of what I've presented today has been more philosophical, this is what it is, but practical examples of how you would do evaluation or how people have done evaluations would be very useful. How this links to well-being, other frameworks, and then being sure that we have that, like I said, practical application. And then finally, this is our selection of task group members. I'm very excited at the diversity we have here, both in career stage and in geographical distribution and area of particular focus, whether that is industry or regulatory or research. And you can see the map behind is actually the countries we have represented in this task group. Thank you.
Thank you, Nicole, for this very complete presentation. And so with this last presentation, I think we have to thank all the speakers and the co-authors for their very informative presentations. And I would like to come to the last Q&A session of discussion. So we still have half an hour approximately for questions to all of the speakers from the two sessions, first session before the coffee break and the speakers right now. So I would like, maybe we can thank the speakers of the second session, and with this I propose we move to the Q&A session right now. And so we invite, of course, the speakers of the first, they are hiding somewhere here, but they have to come on the stage, and we will move to the room.
Okay, so just to explain the procedure, the co-chairs now left the podium, because the major figures are those who gave the presentations. And you are supposed to ask them questions, so it's not our job, but it's their job to answer. And there was not enough space, so we really had to leave. So please now, yes, Alexander, start the discussion, please.
Thank you, Werner. Alexander Ulanovsky, Committee 2. First of all, it's my good chance to congratulate all the speakers today for the extremely interesting and valuable talks, very thoughtful and very well delivered and contributing to the good thinking, I would say. And in relation to the first part of this presentation and talks given, I would like might be rather not to ask, but rather comment on few things which we have heard in the part 1. This is related to some inference which is derived from the excess relative risk graphs which were shown today. And we always should probably remember that the excess relative risk is relative quantity relative to the baseline incidence rate. And that means that in some situations when the baseline can be affected by other factors, by competing risks, then our judgment based on the excess relative risk can be not very well established. For example, we have seen the interesting figure which Richard showed us comparing the MAIAC, all solid cancer excess relative risk versus LSS, where MAIAC figures were lower. And then immediately the question came to my mind saying that could it be that the baseline incidence rate in the cohort was higher? Or could it be that there were some competing factors which reduced the outcome of the incidence of this endpoint considered in the cohort? I mean that some non-cancer effect like the cardiovascular diseases, cerebral vascular disease, or whatever. So, and another example which could be given in this with link to this situation. It's well known if one, for example, considers atomic bomb survival cohort and analyzes the risk of cancer of uterus, one will find very different excess relative risks for cervical part of uterus and the remaining uterus. And the answer is because this in cervical cancer was known to be tightly linked to the HPV occurrence, and that's why these two different parts of the same organ displayed us a very distinctive excess relative risk. However, if you would see the excess absolute rates, they for these both parts, which are attributed to radiation, they appear very similar. So that's why it's rather not a question, but probably invitation for the discussion on our interpretation of the epidemiological data. Thank you, and sorry for taking that much time of valuable time of everyone. Thank you.
Yes, thank you, Alexander. Yeah, you've touched upon a very interesting topic, which we could speak about for quite a while. Funnily enough, in the first ICRP symposium, I spoke about interactions back in Bethesda. And Angers touched upon this when he showed that slide about how risks are moved around populations, because the way things work at the moment in publication 103, the risks predominantly come from the lifespan study of the atomic bomb survivors, mid-20th century Japanese population, of course. And the problem then arises as to how the risk estimates that you derive from the atomic bomb survivors. So you have two models. One is the excess relative risk, which is just the proportional increase in risk relative to base background, and the excess absolute risk, which is just the additional risk on the top of the background. How do you move that around populations with different baseline risks, as you said, because it very much matters to the results as to how you do that. Unfortunately, knowledge of how to do this under most circumstances is rather limited, which is why you saw in Ajay's slides the difference between the ICRP recommendations and the BS7 recommendations. In general, there is some common ground, and that basically comes down to a lack of knowledge about how radiation might interact with background risk factors, and that comes to your point about the MIAC workforce, for example. What I showed there was the comparison of the dose responses in terms of the excess relative risk, i.e. the proportional increase in risk, but you have to take into account the background risk as well, in as much as there might be an interaction between, let's say, smoking and radiation as far as lung cancer risk is concerned. So, yeah, as I say, you've touched on an interesting point. It is something that that can be expanded upon quite extensively, and for sure that we are going to be dealing with that for the next set of recommendations. And you're a part of the task group that's going to be looking at part of that as well.
Could I add something to this? Yes. So thanks. That was a very interesting question indeed, and Richard answered it. But I think there is another dimension to this problem, and that is that, as you have shown, the situation is very complex. How can we simplify it? We have been hearing a lot about we need to simplify the system, and we need to be better at communicating people and, explaining the problem of risk. But there you are. It's complicated, and we heard also today morning in the keynote lecture that our system, that's how I interpret it, our system of radiological protection is to be blamed for the fact that people are afraid of radiation. Well, and I would like to now use this opportunity to kind of defend the system and defend us. The situation is complicated. And we can't make it simple because, we should try, of course, no question, we should try, but it's complicated. So maybe we should become better in educating people. How educating? How? Well, in showing them, look, this is complicated, and what can we do about this?
May I just add one? So, for example, when we compare high-dose rate exposure, LSS, with low-dose rate exposure, let's say MIAC, this was your example, then we say, okay, this could be an indication of a dose rate effect. But we should keep in mind that the baselines can be different. So this is something, I would say contributes to the uncertainties. And we should always keep this in mind. I just want to make one thing. I fully agree that it's so complicated. There are so many parameters, but we should also not forget that we have already more than 120 years of radiation research. Since 1895, when the X-rays were discovered. Since then, we are doing radiation research, and already 1 year later, we have the first recommendations on radiation protection. So yes, it's complicated, but we should also communicate to the public that we know quite a lot already. But I mean, that is science. The more we know, the more we don't know. If we have answered one question, we have 10 others. And in the end, yes, what can we do? So what should we do? Just to work, yeah? Yes. So let's continue working, and part of your work is to ask questions, please. Oh, yes. Denison, please.
Hello. I'm Denison Santos from Brazil. I have a question about epidemiology. I have a question. Okay, it sounds better now. I have a question. I am Denson Santos from Brazil. I have a question about epidemiology. I mean, there is a group of occupationally exposed workers, which are aircrew workers. So my question is, I mean, this is a large group, but they have low doses. On the other hand, they have comparatively high average effective dose, annual effective dose, compared to other groups of workers. So the question is if epidemiology can benefit from the knowledge of aircrew doses for effects or whatever.
Yes, for sure. And there have been a number of studies of aircrew, including collaborative studies. And they are an interesting group, and they will become more so because of the cumulative exposure that they will experience. At the moment, it's very difficult to establish a radiation-associated risk with the aircrew, but as with the nuclear workers, collaborative studies is the way forward on this, and having crew with large cumulative exposures because of long employment periods. So I mean there is certainly potential in there. And the other interesting thing with aircrew, of course, is because they're up in the air, they get more neutron dose. So whether it would be possible to actually get anything out of neutron doses remains to be seen. I would say, however, and what has been experienced with the studies so far, is, how can I put this? Aircrew have unconventional lifestyles, and that can actually be seen in the disease patterns of aircrews, which I won't go into details, but which complicates things.
Just add one small comment to that. I mean, I think the other thing to keep in mind is that, you know, aircrew have significant and prolonged circadian disruption, and disruption of circadian cycles is known to be related to cancer risk. So, it actually adds an additional complication, I think, to what Richard just outlined. They are a potentially very interesting group from a radiation exposure point of view, but they'll be very difficult to kind of disambiguate all the complicating factors.
If I just may, and they are also interesting from the RP point of view because a lot of those cabin crew members are females, young females, yeah? And this is not actually what we want to have in radiation protection, but that's the cohort, no?
So just one sort of thought that came into my head when you said that, Werner. I mean, one could argue in radiation epidemiology, particularly occupational radiation epidemiology, that females are an understudied group.
Hi, Dmitry Klokov from IRSN. I have a question about the detriment. I guess it's for Dr. Ban. I totally agree with some of your slides that there should be a huge effort to make it comprehensible, because I think it's incomprehensible not only for the public but also for experts. Like, I couldn't understand what's the contribution of the cancer effects and non-cancer effects, for example, inheritable effects. So that's my first question. Could you give a bulk number of, let's say we have a detriment of 100 per gray. So how many 100 units per gray. So how many units will be coming from cancer effects? How many units will be coming from non-cancer effects, for example? That's first question. And second question, how do you see, like, what particular actions you can take to make it understandable, the detriment?
This point is contribution. Understandable and contribution of predictable effect. Okay. But mostly about being understood.
So I have not catched your question correctly, but you're asking possible contribution of cancer and non-cancer effects
Relative
to the
contribution
detriment.
of two, yes.
So no answer currently, actually. But so one big question is, as I explained, whether to include, for example, circulatory disease in detriment calculation. If we include circulatory disease, then a significant increase will be anticipated. But of course, it depends on what kind of approach we take, or there could be a conclusion that there is no need to include those kind of non-cancer effects. So currently, there is no answer. So,
okay, so then I have a follow-up question. Because of one of your bullets to make the system based on solid science, the detriment concept to make based on solid science, sorry, because you are trying to make the detriment calculation based on solid science, do you think that there is solid science background to include the non-cancer diseases? Because we, for example, we know that cardiovascular diseases, the risk at doses below 100 milligram are not statistically significant. So do you think trying to introduce cardiovascular disease into the detriment calculation can be called based on solid science in this situation?
So that is because there are many task groups, not only update of detriment, but of course task group 1 to 3 for effect classification. So these task groups are interlinked. So depending on the discussion in other task groups, work or conclusion of other task groups will be affected. So they are interrelated. And of course, we need solid science, and we stick to science, but finally, we need to make a judgment. That is expert judgment. So even if we don't have a solid evidence about increased risk of circulatory disease at low doses less than 100 millisievert, if we make a judgment, it is a prudent approach to do so, then we'll do so.
Okay, thank you.
Maybe just to complement that there is an ongoing UNSCARE evaluation on circulatory diseases, and also there is 119, is it task group 119 as well. So we hope that by the time for the decision of the new recommendations, there will be solid science, at least what science is available to date. Thank you.
If there's no other question, but don't hesitate to ask question. Come on, there are 10 speakers, so you should have at least some questions. In the question by Dimitri Klokov, there was also a question about how to better communicate the detriment, and you spoke about one possibility which is going through DALI. So maybe could you expand a little bit more on that? You or Ludovic, I don't know who is the best one. What do we expect if we try to express detriment in something like the DALI? What is the advantage, and could it answer to the question of Dimitri, how to better communicate, or to better understand what means detriment?
So you're talking about how to express the detriment. So as I explained in my presentation, current detriment, so we get numbers, but convey the meaning of numbers, it's quite difficult. At least lay people do not understand. So one way is, as I said, use another metric widely used in other fields, and DALI is widely used for public health sector and other sectors. And it is at least understandable because how long your life is damaged or disabled as a mathematical expectation, it is quite similar to that of index of harm ICRP once tried. So that is a possible choice.
Maybe another possibility is with regards to the severity aspect of the calculation of detriment. We've been thinking about using daily, for instance, as a way to express detriment. This is something that we're going to investigate. I'm not saying that tomorrow, we'll say that basically the detriment is one daily perceived or whatever, but it may be also another way to speak with people, a shorten of life rather than, a number of case bed, 1,000% gray, and so on. But basically, I think that if you want to communicate with people, you need to sit around the table and to understand the question and to take a piece of paper, a pen, and to discuss with them. I'm not sure there is a magic way to present the detriment. I think Nobuhiko shows very interesting figures on the way to communicate for maybe specialists or so on to make it more understandable. But at the end of the day, if you really want to explain what is detriment with people with no experience, you need to sit, I think, to take a piece of paper and, to start to discuss with the people.
Just to follow up, I think we should understand ourselves very clearly first. What is the detriment, how it is calculated, how it relates to, for example, other risk factors. For example, like I said, if we calculate that a detriment from 1 gray is 100 units, whatever it represents, how does it relate to, for example, background detriment, like background risk of cancer? Is it going to be 400, 4,000? So this is the type of questions I'm having.
Ludovic, sorry, may I answer to Dmitry? Because it was clearly said in the presentation from Nobuhiko-ban that this quantity is introduced for the purposes of radiation protection, and it focuses the low dose exposure, and only as the endpoints, mostly stochastic effects, cancer. And then genetic, in less extent, this is okay, this is under dispute, but again, we could discuss now the concept of detriment for the radiation protection purposes. So the 1 gray example is a little bit inappropriate.
I took it from the slides.
Okay.
So whatever the metric we use, we need some benchmark to make a judgment. So in that sense, so background level, so for example, we evaluate the significance of assessed lifetime risk. We want to compare it with the baseline risk. So in that way, some benchmark is needed to understand and to convey the message to the public.
Can I add something here, Nobuhiko, to what you said, but and also what Dmitry said? It's, yeah, you can compare the detriment from radiation exposure to the same type of detriment from another source, but when we are discussing planned exposure situations. So another aspect of looking at this is saying, well, if you're not exposed, or you're exposed, you have a certain detriment. But if you're not exposed, the consequences, and here the consequences, the magnitude of the consequences or the width of the consequences is outside of the detriment. If you're a patient, you're exposed to radiation, you get a risk of cancer. You increase your risk of cancer. If you're not exposed, you will die of cancer or of another disease that will not be diagnosed maybe. You see, so it's complicated. I think it's much, if we want to communicate, you should include other aspects or compare, let's say, the consequences of not taking an action with the consequences of taking an action?
Yeah, so it's complicated, I admit. So if we calculate or assess just change or difference, then there's no comparable, so we cannot make a comparison. But anyway, even if we quantify a metric, itself does not have meaning, so we need to assess in some way. So try to make a comparison, for example, as a chemicals or other factors. So that kind of comparison will be necessary in any way.
Okay, thank you. I'm Kai from Japan. Yeah, we come back to the origin of the detriment, 1977, Publication 26. At that time, the ICRP defined effective dose. Effective dose required the tissue weighting factor, you know. The tissue weighting factor based on the concept of detriment at that time. Nobuhiko but explained it very detailed. But detriment is not communication purpose, I think. After Fukushima accident at that time, I have to explain what is happening, the risk, to the public people. Media people that say that 5% perceived by ICRP recommended. No, this is about only radiation protection purpose. Communication is but cancer incidence and only look at the even survival data. Yeah, this is like this, like that. Yeah, well, communication purpose is actual data is very important, I think. And the second, this is comment, I mean. Now is current epidemiology seeking the statistical power. But putting study, the meta-analysis, like in works. But a Japanese study, ongoing study, nuclear work study, G episode, not famous, cohort. But this cohort, but very good quality of dosimetry, very good information, lifestyle, socioeconomic status, and very high quality data. But unfortunately, the size, statistical power is not so large, very small. But this is information, this is very important to communication purpose, because comparison other lifestyle risk, for example, smoking and lifetime disease, and recent paper provide the non-cancer data. Of course, 2 years ago, the cancer risk comparison. And maybe I think my conclusion is that, epidemiology study, of course, is seeking statistical power in works at a big database, but a very small database, like J episode in Japanese, like nuclear work study, this is very important information for communication purpose. This is my message. Okay, thank you.
Is your comment on the same topic? Yeah. Okay, so briefly, I would like to emphasize the fact that for the implementation of the radiological protection system, we need to select radiological criteria. And then for selecting radiological criteria, the detriment, which is an indicator of risk, which is a construction, is useful. This is not the only element, but this is quite useful because if there is no risk, why to select a radiological criteria? So for me, there is a need to have an agreement on what is encapsulated into the health detriment. We need to have, we know that we have value judgment behind that, but at the end, this is something which will allow not to select one radiological criteria, but to help in the selection of radiological criteria. So with regard to that, all the different development on the detriment are very useful, and then is it only one or several radiological detriment that could be discussed, but at least we need an indicator of risk to select the radiological criteria in the perspective of implementing the radiological protection system. And as it was mentioned previously, this is really in the purpose of radiological protection.
So I think we are at the end, so it's going to be the last question, and we're going to close the session after that. So please go on with the question.
William Stewart, Canadian Nuclear Safety Commission. First of all, I'd like to thank all our esteemed colleagues up there for the presentations. I thought it was fantastic. This one comes, again, it's to the same presentation, and there's a slide on that included information on breast cancer, and it included that the assumption that male breast cancer is equivalent to 0 or a non-factor. And I'm wondering if we're doing ourselves a disservice when we actually present that. Male breast cancer, although quite a low factor, is a factor of 1% to female breast cancer, but that is mostly genetically carried forward. The actual radiological induced or estrogenesis induced breast cancer in males is actually a higher percentage. So by including a slide where we're saying that that is considered to be a near 0 value, is maybe doing a disservice to ourselves and the population and the doctors that are dealing with the information, including that male breast cancer is a factor that needs to be considered because it's less likely to be genetic and more likely to be radiologically or environmentally induced.
Yeah, not my slide, but I can say that male breast cancer has certainly been studied in the atomic bomb survivors, and it will be included in the, for example, in the worker studies. So these are included. The problem is numbers. So if you need the numbers to have the statistical power, which of course is much more common in females than in males, but it will be included in there. And of course, in the nuclear industry, at least historically, there are more males than females as workers, particularly getting higher doses. So, these, as far as I'm aware, I can't quite remember the details now, but I don't remember any statistically significant excess of male breast cancers, but the numbers will be collected.
Thank you, Richard. I think it was the last word, and so there were many questions on the detriment, and I really think we have to. It will be the occasion to come back to that to next symposium. But for people that have questions on the detriment, I would like to remind that there has been publication 152, which has been published 1 month ago, and you can find everything. I hope it's really much clearer than it was in the past. It does not mean that it cannot be improved in the future. So with this, I would like to thank again the speakers. So I think we can, all the speakers.